and then that was pretty much near the end, like a week ago, so crazy just to think about how far we've come. Yeah. Do we do slides? Okay. So first, we wanted to take a moment to acknowledge the land that we work on. So the ONRC acknowledges the indigenous peoples on whose homelands this research is being conducted, including the Macaw tribe, the Quileute tribe, the Ho tribe, and the Quinault Indian Nation. We have a responsibility to improve relationships between nations and to improve our understanding of local indigenous peoples and cultures. Next, we'll do a quick introduction of who we are. We can go over on screen.
I'm Drew, I'm the third year at the UW.
I'm Delaney, I'm also third year.
I'm Margot, I'm going into my senior year at UW. Trash.
I'm Kyle, and I'm also going to be, I'm going to my fourth year at UW. And then Courtney, as you've already been introduced to, is one of our wonderful supervisors, and Eric was our wonderful crew lead this summer. And then you'll see pictures with Nate and Lizzie who are at the bottom there. They were with the Doris Duke Conservation Program, so they left early this summer, but we still wanted to include them because they were a big part of our crew for most of the summer. Yeah.
Right, so this is just a brief overview of the summer. To start with, we did the ethnoforestry study over in the bush, and then we moved on to LTEF, which is the long-term persistent plant TV study in South Dakota. Then we moved over fully to the type 3 watershed experiment area, did some soil sampling, then electrofishing, and pre-treatment. Thank you. So yeah, I'm going to talk about the ethnoforestry study. As I mentioned before, it's in the push. So this study has been led by Courtney, as you just mentioned a few minutes ago, and ethnoforestry is a people-focused forest management practice, and this specific study is aiming to incorporate local knowledge into state plan trust timber harvest, study the impact of including understory plants in timber planting, and promote angler habitat, which is essentially anything. You can see an example on the right. We have some pretty cool about that. Some various so yeah, a brief overview. This study aimed to incorporate local knowledge for cultural and personal use of plants and access to land for hunting and fishing. And we study a lot of understory plants to better understand the ecosystem health and natural regeneration. And see how they have effects on timber growth, and then support other populations. So we had 3 treatments. We had agroforestry one, which had dense understory, early sterile, which was spaced out understory planting, and then our control had none. And the subtreatments were the amount of slash, which was either low, medium, or high. And wildlife exposure levels, so fenced-in plots or no exclusion and no fencing plots in that in some ways we have covered. And as for the timber portion, the controls have 360 acres or trees per acre of Douglas fir, no red alder, early sterile with 180 trees per acre of Douglas fir, and red alder at 50 trees per acre, and agroforestry at 180 trees per acre of Douglas fir. And so the goals were observe the relationship between understory growth and timber growth, wildlife effects on plant growth, and ensure the efficacy of novel treatments for. And so if you were an ONRC intern at the ethnoforestry site, you would be doing a lot of understory plant ID. I know I can speak for more than myself when I say I learned a lot. About different plant ID methods and identifying really tiny little leaves and seeing them later and having them look like completely different plants. The sapling measurements, we would take the root collar or like the measurement of the base of the tree and height and along with browsing levels, so like if animals have munched on them or not. And there were a handful of planted understory plants that we had to go find in different rows and plots, and that was part of our challenges. Sometimes they'd get pretty dried up by the sun, and it would be difficult to find them, especially under all the slash. But we found saplings, we found little tags, we got really good at balancing, that's for sure. And the weather, because we were in a clear cut, rain or shine, we're out there, you know, always keep your sunscreen and rain jacket in your backpack. It's a great start.
All right, and then our next project that we worked on was the long-term ecosystem productivity study, also known as LTEP, which was in Saco, Washington. So this is intended to be a 200-year-long study. It was created in the early 1990s, and the treatments were implemented in 1995, and it has replicates across the Pacific Northwest. So we were working in the OESF in Saco, Washington, but there's also other sites in Oregon. And the overarching research question that is asked with LTEP is what factors, influenced directly or indirectly by management, most affect the long-term productivity of the land. And the experimental design for LTEP is that there are 40 experimental units. There's 4 blocks of 10 treatments, silvicultural treatments, and in each block there's 3 early cereal, which were clear cut and then replanted with an equal mix of Douglas fir and red alder trees. 3 Douglas fir treatments, which were clear cut and then replanted with only Douglas fir trees. 3 late cereal, which were heavily thinned and is aiming to create old growth characteristics eventually. And we were working in the late plots. And then is also a control treatment, which is well, no treatment is the control. So for the experimental protocol in LTEP, we would locate tree plots using maps and using georeference maps to find the plot corners, and then we connect the plot corners and pick a turning point tree, which is a sort of central tree that we take a lot of our measurements to and from. And then we would measure residual and ingrown trees and note the species, whether or not it was alive or dead, and measure the DBH, which is the diameter at breast height. So we wrap the DBH shape around it and find the diameter of the tree. And then we would know whether or not it's LiDAR visible, which is whether or not it would be visible to the remote sensing mapping. And then also taking the distance and the azimuth on a compass to the turning point tree, which also helps a lot for the LiDAR mapping. And then also we would take note of whether or not there were any anomalies with the trees. So for the experience at LTEP was a lot of being attacked by nature. It was very, very dense plots, but a lot of really, really small trees that are really close together. So a lot of being hit in the face with sticks and branches. And so one of the challenges was just finding the plot corners in the first place, because a lot of them, there was really low visibility in a lot of the plots, and there's a lot of down wood, and just finding the plots is really difficult. And then also with the low visibility, taking the compass readings was hard a lot of the times. And working in those like really close conditions could get pretty claustrophobic. So even there would go like whole days where it would just be like dark and you wouldn't see the sun until you get out of so, and also the wasp ground nests, we had some fun experiences with those a few times, but we made it through.
Yeah. All right.
So then some more just of the overarching goals for LTEP are to address long-term productivity and forest management and to find new concepts for experimental adaptive.
So after our time at LTEP, we moved on to the type 3 or T3 watershed experiment in the OESF. So type 3 or T3 refers to the classification of streams that reside and are studying in this like portion of forest. So a type 3 stream is any fish holding stream or a stream with the right characteristics to hold fish. And T3 is a major collaboration between DEAN and the UW it's striving to incorporate socioeconomic factors into its experimental design and develop new timber management strategies to like maximize productivity while also providing ecosystem services. T3 is studying both upland and riparian systems, so like forests and streams. And then fun fact, T3, along with the Olympic experimental state forest, the logging money that is raised when they sell timber, goes back to schools, houses, hospitals. In the state of Washington.
So there's a lot that went into the experimental design of the T3 experiment. It explored 4 management strategies with a variety of descriptions, which you can see on the slide, but I won't get into all of them. Including 2 alternative strategies. We're studying 2 alternative strategies to standard ways of management as well as it incorporates many treatments, including active restoration and all the rotations and others to try and expand the management toolbox of DNR forestry management within the USR. Because sort of the standard fixed descriptions to timber management can be restricted. So we're hoping to diversify the ways that we forest. And so there are a lot of goals of the T3 study, but mainly looking at people-focused ecosystem management, looking at the health of the communities as an important facet of the ecosystem overall, as well as collaboration. This is one of the first large-scale collaborations between the UW and DNR, and also includes other agencies such as the Pacific Northwest research station with the U.S. Forest Service. And hoping to create science-based active management model, as well as plant descriptions, multi-scale management.
So I get to talk about, I think, the most exciting project, the soil sampling that we did in the T3. And I know Dirk may not sound like the most exciting thing, but there's a lot we can learn from soils, and so I'm very excited to be out there and collecting samples. And so we're collecting soil from the watershed within the T3. And we were getting 2 main measurements from these samples. That was soil nutrient content and bulk density. And these samples are being collected, and this data was being measured about the watershed before the logging and treatments occurred, so that we can compare after we do these treatments and come back and want to see how the soil was affected. And so the equipment that we were using out there were these super long augers. That's the image on the right. And so they're kind of about to here on me, so probably like 5 feet or so tall. And those were used to get the soil nutrient content samples, and so they pick up a lot of soil at once. And then we use these on this other image, a slide hammer, and these help us get a soil core, which we can use to measure bulk density of the soil, which kind of tells us about the water holding capacity of that soil, and then thereby the nutrient holding capacity. And then we use a lot of like big old Home Depot buckets and just some basic trowels. So we had a lot of equipment, but it was a very interesting study. And these are just some pictures of things that we were doing just to give you an idea of what it looks like in action. So down there, this bottom right image is a picture of Josh using the soil auger. And so you stick it in the ground and you twist it. And so it's a great 4 exercise. And we pour our measurements into the buckets, and we end up not using all the soil that we collect. And so I thought this was a cool image of Josh tossing the soil out of the bucket back into the forest. And then up there on the top right is a picture of one of the soil cores that we took. And so they're not super big compared to the amount of soil that we get from the augers, but just as important. And as I was saying, there's a lot of gear. And so that's what the back of our Suburban looks like on a daily basis. And if you can also imagine underneath that, covered in pine needles and dirt. So we'll be getting a nice good clean at the end of this week. And the T3 is very, has a lot of terrain. And so we were hiking up and down these hills with all this very awkward and large equipment. And this is one of the nicer slopes, I think, that I managed to get a picture of. The ones that weren't as nice, I wasn't focused on taking a picture. So, yeah. So at each plot that we get to, we set up a plot center and we collect soil in 3 spots kind of around the center, kind of in like a circle. When we collect at 20 centimeters deep and 50 centimeters deep at each hole. And at each depth, the soil is mixed together so we can get an accurate representation of that whole plot. And then with the slide hammer, we do that in the center of the plot to collect a bulk density sample at 20 centimeters deep and then at 40 centimeters deep. Again, so we can get a broader idea of what soil looks like from the top of the soil all the way down to pretty far down. And so we collect half a gallon of soil from each depth for the content, the nutrient content, and then we collect about a quart of soil for the bulk density. So we'll end up with about a gallon and a quart of soil at each plot. So you can imagine that adds up. So the main goal of the soil study, the T3, is to understand how forest management affects soil health, mainly the composition of carbon and nitrogen, along with other soil indicators or health indicators. And as Josh mentioned, one of the goals of the T3 study is to see how incorporating alder in affects forestry management practices. And alder fixes nitrogen from the atmosphere, it increases that soil nitrogen content. And so we want to see how these treatments are affecting the soil nitrogen. And soil health is linked to tree health and productivity, as well as nearly all other ecosystem functions. So it's not just the dirt that we walk on, but has a lot more going on down there. And so from our perspective, we like to lovingly call it, we were just out there in the movie Holes. If you've ever seen that one about the prison camp where they have kids just dig holes to build character, and we were just out there digging holes. And it was more for building character, but thought that was a fun little comparison. And some of the challenges of soils, the soil sampling were definitely, I said, carrying that heavy and awkward equipment, and also carrying all of that soil up and down those slopes. By the end of the day, we have around 40 bags of soil, which is probably around 40 gallons each, so it's quite heavy. And as you're going down into the soil, you can imagine that there are roots and rocks, because that's where the plants and the rocks live. And another big challenge that I faced quite a few times was dropping a bucket down a hill, and you kind of drop it and then watch it just like keep tumbling and tumbling. And you kind of have to just accept it, you have to go down again. But on the flip side, we saw some amazing views of the watersheds, and different than other projects, we weren't collecting data, so there wasn't as much need to like really hear each other. So we were able to listen to some music, which is very nice. And it's a great leg exercise. Our legs are shredded now, so it was all around a good time. And so those are kind of two of the good little views that I found. I don't know if that video will play, but that if it does, let's see. And I messed it up. So it's okay. Basically, though, we just kind of like would fill our chem lab with these bags of soil, and like every counter space that we could find was filled with soil. So it definitely was crazy to see. By the end, we had around, I think, 500 bags of soil. We'd have to take it all out and dry it so that we could process it. Very cool. I don't think that is. Sorry. That's okay.
So after our work at the soil, we moved on to upland tree monitoring at T3. This involved a lot less gear, so it was quite the break. So the tree monitoring is just observing the positions of individual trees of 2 species of interest, which were red cedar and Pacific silver fir. We were tracking these positions a lot like we were at LTEP, with just a distance from a central starting point and a compass azimuth that like that. The upland monitoring is trying to, or is, getting pre-treatment tree heights using LiDAR canopy height models. So we know where these trees are, and we have a bunch of remote sensing gear. So we can just like find the tree and then look at the computer and it'll. And then we're also using this data to improve species identification of computer programs. So in a LiDAR model, a computer might be able to identify tree species based on how they, and we can provide the data we collected as practice for learning computers. So now I'm going to walk you through a day in our life at the tree plots. So we find a plot or an area that's just pretty dense with whatever species we're looking for. It was mostly Pacific silver. Then we would pick a turning point tree that was probably hopefully super large and visible in our LiDAR models. And then around this tree, we would number all the trees with tree spray paint that fell in our 17-meter radius. And then for every single tree, we would get species, diameter, azimuth, and the LiDAR visibility in this. Overall, after like 3 days, we counted almost 200 red cedar and over 200 Pacific silver. And then as a bonus protocol, while we were doing that, a small, like 2 people broke off and did some understory coverage plots. So 10 meters away from the turning point along the northwest and southeast directions, we would make little 9-meter square plots and just identify all the little plants growing and get their ground coverage estimates.
Sometimes we had up to 22 species within that 9 meter square. Very diverse.
And the final project that we worked on was electric machine. Mr. Stearns in the back. So essentially, we're doing this measuring fish population. To take a variety of measurements, fish length, weight, anomalies, as well as temperatures, like water with the goal to collect data on salamands and T3 stream habitats. So once again, you get to live inside the mind of an intern. So we would start. So our protocol was essentially to section off 100 meters stream, and that name electric fishing close to what it actually is. You can see the electric fishing backpack, which would carry along with 2 probes, the cathode and the cathode, and we would shock the wire and temporarily snap the fish. We would scoop them out, and while we were doing that, which takes a very long amount of time based on the stream, we would use a variety of different tools to collect that. Some of them seems really much bigger, to track that. They get us a good idea of what this is like. So electric fishing came with a lot of challenges, including a common theme was the cheap which was the terrain. Steep, slippery slopes that have to go up and down and pull over gear, as well as then sharply along the streets. But this time with the special added bonus of felt bottom boots, which really taught me to appreciate the friction between my shoes and the ground. We also faced some dilemmas on socks or no socks, which I'm sure some of you might have thoughts on with layers. There is a correct answer, but there is not a ubiquitous. And another problem is that this is all of our first time doing electric fishing, and a big part of that is seeing how far down the fish is. So our very patient supervisor, I'm sure internalized some frustration as our nets just went right above the fish over and over again. And then something I didn't mention was the buzz which got the intense. But overall, electric fishing was a very rewarding experience. It added some zest to our lives. Yeah, great time.
Right, so to talk a little bit about our life at ONRC works, because we have 4 10-hour days, we have a 3-day weekend every week. It's gonna be really hard for me to go back to weekends. But in that time, we did so much. You can see all the hikes listed. I feel like my favorite was Storm King. It was quite the scramble, but it was so worth the view. We spent a lot of time in Port Angeles, which we came to know as the big city here. I'd never been to a Walmart supercenter. That was big for me. We got very excited about, the college budget, which shopping we can do after for grocery. We spent some time camping over in Bear Creek and Shiny Beach, which was really fun. We surfed in ocean shores. I had never done this. So that was quite a new experience for me, especially in the very cold waters of the pier. But that didn't stop us from swimming all over as well, doing some bonfires. Yeah, slide, we'll see some more pictures. We competed in the second annual Forks pickleball tournament on 4th of July. You can see Nate and Kyle in their national uniforms. They did very well. I got a sportsmanship award. And we have our creation from camping, camping cuisine of spaghetti hot dog, which is the spaghetti dog. That was very fun. And yeah.
Yeah. Having to cook 8 different meals in a small kitchen every day after a long day for breakfast. Yeah, so collaborated and that's really good time. That's some good bonding over cooking.
Fortunately.
We've also had a lot of friends hang out and play some sports together out on our facility. As well as we really had a great time in the local culture with the variety of events that folks have for us. Employee days, the 4th of July events, corn, boil, and branches. And finally, what I think is our crowning achievement that we mentioned too, we are officially first to finish all 5 meetings. Yeah, we just really had a great time experiencing famous forest hospitality.
Our most important project of the summer was taking pictures of Drew. These are mostly pretty much all my images. I made it my mission to take a picture of Drew every day. Wasn't every day, but I tried to catch him when he was most off guard. I think one of my favorites is that very top right one where he's all muzzled up. It was our like first day in the rain, and it looked like when moms put like the giant puppy coats on their kid to go out in the cold. But I did make it my mission to do my daily Drew picture. So I feel like I had to include this because I was really proud of my collection. And then finally, we just wanted to say to the people in City of Forks, the ONRC, thank you so much for having us out here. It's been an amazing experience. And to Eric and Courtney, thank you so much for being awesome supervisors. Everyone that came out today and on Zoom, thank you so much. And we can take questions if y'all have any.
Maybe it's more, should be directed more than support, but when you're taking soil samples, if you're not, if you're thought to surveying the microrhizomal biome, since you're digging down, presumably have an effect on what's above ground, but has there been any thoughts towards that?
I'm gonna push this over to Bernard, who probably has more sense of this.
Yeah, no, the below ground biology is very interesting. Of course, you can't really see microrhizae, they're too small. You run into a lot of roots, I'm sure they've got some roots. But yeah, no, we're interested in some new techniques coming along. Including there's some DNA. We hope to get more and more into that. So this is something about how certain trees share microrhizae and more and more.
Interesting.
A comment and then a question.
I think they ought to invest in their forks. We're running up and down these hills, and loggers wear it for a reason.
Yeah,
The traction is a lot better off. Wouldn't be cheap.
maybe a worthwhile investment though.
And you can't order them when you're going to the grocery store.
That's right. At least not, nobody they did.
We saw the sign at the.
Yeah, right.
Yeah. And also I saw on one of the slides, a cedar red alder polyculture. Normally we talk about fir and alder, but that was specifically something about cedar. In polyculture, what is there anything significant to say about that? We respond well, and know that the alder really promotes the wellness for it.
Yeah,
idea, I think, is that since cedar is so slow growing, those would be a 16-year rotation, and that kind of include 2 rotations in
Yeah.
30 years. So I think it's from a productivity standpoint.
Yeah, the combination of the slow growing shade tolerant cedar with a fast growing sun dependent red alder was an interesting combo for us.
Last year when we went to that food pit down Clearwater, that's what I call it. There's somebody who was talking about the idea of planting spruce and cedar together to discourage the elk from browsing on the cedar. They're gonna eat it. Of course, the spruce grows so much faster. So that's gonna be a very labor intensive process to get. Someday, somebody's gonna have to follow that, and have to cut those spruce
yeah.
away, so. I wonder if is that a really practical idea? So there's actually some tree study that has, most of the one of them is cedar browse. And so there's a group who are meeting just a couple of last week. That, and they're developing some small scale studies to look at the plantings, spruce and cedar, and the same hole, and a variety of other techniques. Because one of the big concerns with cedar is the deer don't like it. They don't eat them. And so we're trying to figure out some ways to get them to survive. But you can join if anyone's interested in that question. It's a really group you can join and get involved. They're actually the group is designing a study on their own. ONRC going to find it out. And say,
That's a good plug for all the learning groups. So we have a variety of learning groups. I think we have 8 or 9 in total now, spanning a number of different topics. And we have a DNR facilitator that's organizing them, but it's made up of community members, stakeholders, tribes, people from industry, environmental groups, and beyond. So we have a history group that Bernard's really involved with, a remote sensing group, economics and operations group, cedar browse, and a whole bunch more. So if you're interested, I'm happy to talk more about that.
So when you're talking about the LiDAR and such, I know that infrared is used on satellites to see like the density of forests and other plant matter. Is it something like that? I don't know if they use LiDAR specifically for that, or is it more of a ground-based type system that you find kind of in the phone for scanning?
It's a little bit of both, I think. So there's a combination of like aerial LiDAR, which is a little bit similar to what you're talking about, but then we've started to introduce drone LiDAR recently, which is using like smaller drones that fly lower down, which can get more detailed 3D images, and it kind of does similar to it bounces light off the trees and builds that height model. And then what we're doing out here is taking data on these trees that we're getting the aerial information on, and we're like making sure that it's like correct and accurate and like consistent with the lineup.
They're taking in Russian satellites and can conditions you can get.
It can hear within a couple centimeters. Really,
We have to run in for at least 30, Forest Service says 15 minutes, meeting for 30 minutes, and we have a column on our data sheet where we say, and the slide are visible, and the hope is, or it's been done and it's continued to be improved, but the point cloud will take out just the very top part of the crown to determine its emissions.
It's like, if you were to imagine laying a blanket across the top, across the canopy.
Yeah.
And it creates this, you can see, you know, where the high points are, and then that's how you start to identify where.
All right.
How close are you to having enough data points to have it be useful for the machine?
So we had a handful of flights flown last year, and we had ground data taken in both trees and understory. One of our capstone students, actually a last year's intern turned capstone student, worked with some of our collaborators to put together machine learning models, and we were able to use it to identify Douglas fir and hemlock. And I think our accuracy is 92%. So it's continuing to be improved. The hang up was that we didn't have enough ground data for red alder, hemlock, or silver fir. So the crew this year targeted those species so we can improve the model with it. So for any instance where it found silver fir, it would either misidentify it or it couldn't identify it because we didn't have enough in the model to train it. But hopefully we're thinking that 200 trees will be good enough to train it.
It's gonna be a challenge because it's probably gonna become a lot of the elevation. And that would be machine challenges.
I think it's
know where you're actually looking. Pacific silver for the rose, typically rose in higher elevation. And the places hemlock.
You know, with the first year, like, we were lucky to get 30 over the course of an entire day, and then one day last week, we were just like beyond giddy because we got 70 or 80 in one day. We found a beautiful patch and all of them were super visible, and there was nothing, but it was very low elevation. It's like when the first left off the home main line that doesn't have a gate, is where we were able to find all of that.
Yeah.
I think we had a question on Zoom. Brad Harrison had your hand up. Brad, do you want to unmute and ask your question if you still have one?
Sorry. Can unmute.
Yeah, we can hear you.
You can hear me?
Yeah.
Oh, great. Hey, I figured it out. Hi, I am Delaney's uncle, and I used to work for the Forest Service in California doing fisheries. It's been a long time. I'm in Wisconsin now and teach environmental science. But I was curious, like, is there anything that is helpful in terms of understanding the relationship between tree species and soil erosion capacity, like per species per height of tree? Like, is there an indices where you can say that the ecological value of that tree in terms of retention based on slope, I guess? I mean, there'd be other factors, but I wonder what the role of species, diversity has in preventing erosion. And also I was curious about if you guys have been inspired by, is it the Hubbard Ball study, I believe? Is that the right one? Long time ago. This kind of reminded me of it, where they clear cut a watershed and then left another one unclear cut it. Anyway, those are my questions. I don't know if kids can answer them or who can answer them, but.
Yeah, please.
That's pretty interesting. Yeah, actually, I think you're talking about Hubbard Brook. It's in
Yeah.
New Hampshire. Interestingly enough, my father was the founder of. But anyway, in terms of your species in soil interaction, we do know certain species have deeper roots than others, so they could play a different role in preventing landslides. Maple, by the way, is probably one of the deepest rooting trees that are common in this area, and I think they do play a particularly important role in reducing land slippage of soil. On the other end of the spectrum is hemlock, which tends to be very, very shallow rooted. So when it blows over, the root logs are kind of thin. So they do play different roles. And so does the understory, you know, after a harvest, the plants that come back right after harvest are really important. They get their roots down in the soil right away. And I know a lot of people don't like these kind of weedy looking plants that come in to clear cuts, but those plants play a huge important role in getting, stabilizing the soil, catching nutrients that otherwise might have leached down below the surface.
That, I think that's, or correct me if I'm wrong, but that's part of what is special about LTAP is production forests have historically excluded early cereal and late cereal understory species, and this allows for us to compare.
Yeah, I think most of what the interns have worked on in some capacity has dealt with early cereal forests. And I'm a huge fan and I'm super interested in understory, especially in early cereal environments. And it, certainly plays a huge role in, tons of ecosystem processes. I mean, of course, food for ungulates, but also sort of soil stability and and so much more. Any other questions? Oh, Kyle, you're amazing, he says. Great job on an important topic.
Have you unshared, we see everybody who's on your.
I think they're, so thank you for coming. Yeah. Yeah, you folks on Zoom now. Yeah, any additional questions?
Well, there was I think I saw a question was about.
Yeah, or maybe it's on the other.
Oh,
Identity.
yeah.
I guess we have to turn it cameras.
Other.
Well, while you're looking at that, I wanted to thank everyone for coming out and thank Brittany and Eric and all the interns. Huge amount. That's very important. And thanks to Tom Rosman here too, who's our evening 12 benefactor. And I think we should give them a round of applause.
Yeah, thanks again, also to Brittany and Tom and all of, again, folks in ONRC for giving us this awesome opportunity. I know I speak for all of us in saying that it's been super amazing experience, even not just within like our areas of study, but just getting outside of the city and getting our hands dirty and being a part of something a little bit bigger than ourselves, which has been a really amazing experience. Thank you. Who's coming back next year? We'll find out.
All right, thanks everyone on Zoom. I'm gonna end the session.